And then, give me a second. Okay. Okay. Hello, everyone. Thanks for coming to last colloquium of the spring. So it's my great pleasure to welcome Professor Ellen Lindsay to the IBS BIMAC colloquium. Professor Lindsay is currently Associate Professor of Applied Math in Notre Dame. His research uses the mathematical modeling and analysis to understand the role of randomness in biological function, with particular emphasis on PDE, stochastic process, asymptotic analysis, and computational method. So his work spans a wide range of topics, including diffusion process, persist time theory, rare events, reaction diffusion system, and data-driven mathematical modeling with application to immune signaling, chemoreception, and biological development. So today, he will give a talk titled Mathematics of Diffusive Signaling. Please join me in warming welcoming Professor Ellen Lindsay.
Well, thank you very much. Thank you. First of all, let me just say what a pleasure it is to talk to everybody. Just feels like not long ago I was out at IBS 2 years ago at the SMB meeting,
Mm-hmm.
and gosh, what a time that was. And I was near the bakery, and I had lined up in the bakery every day with my umbrella to get all sorts
성심당.
of treats. And yes, that's right. And so much great mathematics going on out there, and and the banquets were a lot of fun. I got to meet Sai and or saw the Sai in person there, and I think next time you guys host, you have another meme. It's K-pop Demon Hunters, so I'm sure we're going to have a whole different entertainment structure next time you guys host. But those wonderful memories. So it's a real pleasure to talk to everybody here, and of course, as usual, just want to mention all the people I've worked with on these projects. And this is actually, now that I'm looking at this here, I'm just, it's insufficient. There's more people who've worked on this. Lots of students missing here, new faculty collaborations happening all the time, and fortunate to be supported by the NSF. And just let me start to mention some broad themes that I'm working on in the last few years. And uh, it's basically this role of stochasticity in biological function. I think we're all thinking about this. It's so important. It's a beautiful conundrum to try and figure out how do we get these reliable deterministic systems, despite the fact that we know there's all these variations and variability, trying to push them off course. So how does determinism, reliability come around with all the variability that we know is out there? And of course, the traditional paradigm is that, there can be an underlying deterministic mechanism and there's some kind of robustness around it. So there's some kind of central clock around everything, and then there's noise that kind of that can be adjusted for. In reaction diffusion theory, kind of one of these ideas is the idea of bistability. So you have some bistable curve, like the S-shaped bifurcation curve. And then if there's a small amount of noise, you get pushed off in equilibrium. But if the noise gets larger, you could be pushed off to some new state. And that would be how the switch worked. But there's also a paradigm that I'm exploring where randomness can also be constructive. So it's not merely some nuisance that systems have to adapt to and be robust to, kind of like how an engineer would build a bridge. Well, I don't know what the wind, or I don't know how many cars or what the wind's going to be in a certain day, but I'll build in some bandwidth. I'll make it this extra strong so that I can so that it can withstand all these different circumstances. It's not like that, right? The noise can actually be constructive. And three kind of problems that I've worked on this pursue these ideas, and one is immune signaling. So this handshake between the T cell and the antigen presenting cell. Uh, and then basically, there's in one perspective, there's a binary decision has to be made. Is there a pathogen or is there not a pathogen? It's a yes or no. So there's a switch has to happen there. How does that happen? Chemoreception, this idea where cells have to make a decision on their environment based on the arrival of diffusing molecules. So there's a release from some agent or point, and the signal diffuses around, and it's received by some agent, and the decision has to be made. and what are the sort of errors that can be made in that process, and also intracellular transport. Is cargo shuttled around the cells and inside cells or around cells so that it gets to where it needs to go, uh, so that it can perform its function. And here's two examples of this directional signaling process here. And there's two different times, two different organisms, two different timescales. So on the left, you see yeast. And yeast search for a mate, so they have a sex. So the males have got to find the female. So there's this extra portion. So the point is, where should I go to find a mate? And what you see at the beginning of this experiment is that the red cells up there and then the pink cells, they acquire a directional estimate of where they need to go. And it takes about 2 hours for them to do that. So 1 to 2 hours is the time scale from where they're sitting there. They're receiving molecules from all the agents around them, and then they're making a decision. All right, this is where I go to find a mate. And that bright portion there. My mouse is working here. It's not. Okay, is my mouse working?
You're
Okay,
not seeing the cursor yet.
okay. No, it's okay. Um,
Yeah.
okay. And on the right-hand side, you see a totally different timescale in neutrophil. So these are some classic experiments, and what's happening here is that the experimentalist is watching a cell at rest, doing nothing, and then at that white dot there, it's emitting a chemotactant source, like a little dot with a pipette. And within a second or two, the cell knows exactly where it is, and then all these downstream machineries kick in, and this is the work that mechanical biologists do. Is like, how does a cell actually exert a force on a substrate to move in a certain direction? And you've got actin networks that activate and all this. But what I'm interested in, what I'll talk about, is some mechanisms like, well, how does the cell know it's there so quickly? Within a second, it knows exactly where to go. It's that the source is immediately to the left, and then it moves towards it. And a lot of these sort of problems, there's a lot of commonalities to them. There's some source. In the pipette example on the right, it's really just a point source. Whereas on the problem to the left, you could think about the whole cell being a source here, and it's emitting some things. But there's some signal or some signaling molecule, and it's absorbed at the opposite cell at some cell surface receptors. And then once that information is received at the cell, there's a decision made about whether to move, whether to react, and if so, in which direction. Mathematically, I want to break this down into two sort of problems, and we'll see sort of treatment of both. There's a forward problem where, and that's what we classically think about in sort of differential equations, is like, all right, we have a differential equation, we have an initial condition. What are the signals received by the cell? Once all those things are established. And then there's the inverse problem, which is what the cell is actually solving. It's like, well, I have the fluxes, I know that I measure the solution, and now I want to say something about my environment. So this forward problem is what a lot of our techniques are geared towards. The inverse problem is what we want to do to try and say something about, give some mathematical insights on what's happening in these problems. Here's another problem that's related, and it's about transport in cellular environments. So, the nucleus is most cells have one nucleus, and we're interested in the transit of materials from the extracellular cytoplasm to the interior. Is that regulated? Well, it's regulated at these gates, these channels called the nuclear pore complex. They're uniformly distributed around on the nucleus. There's around about 2,000 of them, but they occupy 2% of the surface area. Now, these things are very complicated. They're one of the most complicated proteins we know. They're very selective about what goes in and what goes out. They're really a remarkable object. If you don't know about the nuclear pore complex, it's very interesting to read up on again. But mathematically, how would we talk about or devise some framework for discussing transport processes or reaction processes at membranes like this that are covered in numerous small sites where reactions can potentially happen? What sort of tools do we have to describe that? And what about, and a lot of these cases I've talked about here are where there's homogeneous diffusion. It's very simplified, but the reality is that there's cells a very busy place, and one of the most important cellular structures that's in there is the microtubule network, and it's a highway structure which allows cargo and molecules, signaling molecules, to be transported much more efficiently. What you
Mhm.
come to learn when you study diffusion a lot is that it's a very weak mechanism for getting things places. It's
Mhm.
passive, so you don't need a lot of machinery, but it's quite weak at exploring spaces. And a lot
Mhm.
of biological systems have evolved to bypass this with certain linear structures, and microtubules are one of them, so that particles may diffuse freely around the whole cell, but when they become close to some linear structure, they bind to it, and then they undergo a one-dimensional diffusion along this, and they can move towards the plus end and towards the negative end, and there's various motors and that are responsible for this. So this is a very complicated problem now because the particle undergoing some mixed state where it's diffusing freely, but then also going through some bias diffusion if it's bound to microtubules. And of course, there's other structures. We've got these linear structures, microtubules, and then there's also 2D structures like endothermic reticulum. And I know this is work that Jinsu's done, uh, uh, trying to understand dynamics that can happen here, where there's three-dimensional diffusion, but there's also processes happening in two-dimensional substrates where more complicated things happen. How can we sort of use math to sort of understand the roles of the different geometries here? So three-dimensional diffusion with one-dimensional substructures and microtubules, three-dimensional diffusion with two-dimensional substructures like the ER. And of course, one of the ways we can work out this, and I know Jay Kuhn's done some work on this, is think about diffusion as not being a constant. And
Mhm.
of course, it should vary through the cell, in which case you're going to arrive at differential equations that have different structures. And how do actually we deal with non-constant diffusivity is it's very tricky actually. There's a lot of different things to think about, and this is what I'm interested in. So let me give you some mathematical overviews, and of course, some of the things we mentioned there. That talking about diffusion, the dimension is very important. So
Mhm.
1D diffusion is very different from planar diffusion.
Mhm.
Now, of course, we're interested in three dimensions, so we want to go up from three dimensions. And a halfway, a stepping stone is the half plane example. Why
Mhm.
is that? Because you're in three dimensions, but there's no geometry. So everything's assumed to be flat. And that plays a big role, but it's a stepping stone to the full three-dimensional problem that we'd really like to say something about, which is particles diffusing in some three-dimensional region. It could be inside a cell or outside a cell where there's localized reactions happening. These reactions could be receptors where things bind and they're signaling, or they could be gates that have to sort of traffic molecules going in and out of the nucleus. And that's what I'm going to talk about in the first part of my talk. I'm already blowing through my time here. That's what I'm going to talk about in the first part of my talk. And it's kind of a vignette of these problems I've been working on in nine, oh gosh, I'll tell you the first paper I did on this was 2017, so 9 years ago now. And I'll give you a sort of vignette of the different things I've learned and maybe hopefully I can tell you about that. And then this is some more recent work. This is going to be more of a, at the end, if I've got time, I'll talk about two recent papers where we're trying to give a principled answer for how to perhaps
Mhm.
give systematic mathematical ways to talk about diffusion in circumstances like this, where there's some microtubule network, there's some structure where there's different types of motion happening, unbiased motion, some sort of biased diffusion, or perhaps just drift or persistent motion. How can we is there something we can say about this? I'll talk about that in the second part of my talk. So let me talk about a classic problem here it's motivated, hopefully agree, it's motivated by certain things we've talked about here, where we have a Brownian particle and it's got to reach. It's either on the left-hand side, you see a situation where it has to escape through some small window, and on the right-hand side, you see a situation where it's got to reach some reaction site inside the cell. So on the left, it's sometimes called the narrow escape problem. The particle needs to sort of exit through
Oh.
some small window in the boundary, and on the right-hand side, you see something called maybe the narrow capture problem, where the reactions happen at certain condensates or regions in the cell, and the question is, well, how does the spatial configuration of these influence capture time? It's a problem that many folks have studied. Um, and one of the basic things that you might ask is an optimization, not necessarily an optimization question. I guess that's natural just because we're mathematicians and we want to know, but how does the geometry, the shape of this domain, the location of the boundary windows along the perimeter, and
Mhm.
also the number and location of traps, how does that influence things that we're seeing?
Mhm.
And one way you can study this is look at the mean first passage time. So you could say like, okay, what's the average time it takes to reach one of the sites? What is the average time? And to answer that question, you would study this Poisson PD. It's Laplacian equals a constant. And then where there's an exit, you would put an absorbing boundary condition. That's a Dirichlet condition. And where there's reflecting portions, you would put a no flux boundary condition or a Neumann boundary condition. And one of the quantities of interest, which gives a big description of the whole thing we're seeing as well. Obviously, if my starting position is very close, then I would exit very quickly. So I can look at an average, this global mean first reaction time.
Mm-hmm.
One of the things we can do in asymptotics is say,
Mm-hmm.
well, if the traps are small, if these little windows are small,
Mm-hmm.
then we can reduce this to a discrete problem.
Mm-hmm.
Okay, and it's discrete in the sense that all I need to know now is the locations of the traps of the
Okay.
windows. And it doesn't come up quite as clear as here, but maybe you can see this mathematical object at the bottom is quite simple. It's quadratic form. So it's a vector inner product with a matrix times a vector. And we have this matrix here. It's called the Green's matrix. And its ijth entry tells how the ith component and the jth component compete for information. And it basically tells us something about whether traps are close or far apart, how that's going to affect capture time. And it's related to this item called the Green's function. So this is anyone who's done a class in PDEs hopefully knows about the Green's function. There's lots of ways to think about it. It's related to the fundamental solution. So for example, if you think about Laplace's equation in 2D, you know the solution's a log, this the fundamental solution. But then if you put a boundary around it and ask for a certain boundary condition to be enforced, in this case it's the no flux boundary condition, what that does is changes the Green's function in the sense that you still have the singular part, that logarithmic part, which comes from the fundamental solution, but you also have a regular part which enforces the boundary condition. And for certain geometries like squares and circles and things, we can write down it's just a matter of doing separation of variables and blah, blah, blah. And
Mm-hmm.
then wacky geometry like this, we can have various numerical solutions to calculate this. And this is from boundary element solutions here. So we can access this. So we can start asking some questions. If we start looking at disks, and basically what you see in the case of a disk is that the best location for one is at the center. And then you start filling up the space and rings, and
Mm-hmm.
eventually you have to points at a center ring here, and you start filling up different disks here.
Mm-hmm.
And what you kind of see is that as you fill up more and more points,
Mm-hmm.
you sort of pattern where things want to be equidistributed. And
Mm-hmm.
this were some old results from a few years ago that we were able to reproduce with our method. And we can do this for crazy domains as well. So what we see here is, okay, so at small numbers, we have some preference to be at the geometric center. So we want to be kind of in the middle of everything.
Yeah.
Where's the best place to be? Be in the middle of the room. And then as we fill things up and fill in more, we see a kind of homogeneous curved space, what looks like that. Okay, and we see this for different domains as well. So in this kind of oval, the best place is right at the center. And then as you start filling up the domain, again, you see this picture where basically you want to fill up the space roughly homogeneously.
So Alan,
And it's
sorry for bothering.
oh,
Could you remind us what P, R, and G stand for on the bottom of your
P
slide?
is
Yeah.
yeah, it's this term here. Okay, so it's the term inside the tau. Okay, so
Hmm.
it's the term that you should minimize to minimize the capture time. It's the quadratic form.
Okay. Okay.
Okay, so basically what you should minimize, the positional information, what you should minimize to minimize the capture time.
Okay.
So that's what it is. So the tau, it's basically this quadratic form here. E inner product with G times E.
Hmm. Hmm.
And that's for traps inside. If you do it for boundary traps, what you see is that, and then of course the natural question is, well, how should curvature matter? Or if you have a cell which is a very irregular boundary, does it matter where its receptors are on the surface? Well, if you're talking about just the narrow escape problem, well, we looked at the ellipse is the sort of the smallest perturbation from a circle. And what we kind of see is, and we don't know the answer for sure, this is an open problem to really describe this fully, but what it seems like, and you can kind of see on this And again, I just to confirm here, you want to the fastest exit time is where R is smallest. And it seems to be where the curvature is smallest as well. So at the you've got an ellipse here, and at the peak, at the two major axes, the curvature is highest, and then the minor axis, the curvature's lower. And it appears that there's a preference to be at the point of lowest curvature.
Mm.
Now, when we go to some irregular domain, that
Mm.
picture is a little bit less clear. So I
Mm-hmm.
just chose some random domain here. And again, for the first four, we see that the first four critical points appear to be at where the curvature is the most negative. Now, if you really stare at these two crazy curves here, you'll notice, and you do a really careful analysis, is that the minima do not coincide. So they look close, but they don't always coincide completely. But there is a tendency for the minimum of R to coincide with the minimum of curvature. So we have this open problem. We've got some numerical tools we're working on it, but it seems to be the minimum of curvature is an important point where traps should be centered in order to minimize exit times here.
Mm-hmm. Mm-hmm.
So what about this problem in 3D? And I talked about this problem with the nuclear pore complex. How do we describe transport through something like this? Well, this was thought a bit about in by Berg and Purcell. And they asked this question in the many years ago,
Mm-hmm.
and they said, okay, well, let's look at a steady state concentration outside a spherical region,
Mm-hmm.
and on each of the receptors, there's some fixed value, there's some fixed concentration, and otherwise it's reflecting. Otherwise, the cell's impermeable. And you can ask, what's the flux into this, the total flux into this object? And if you do the divergence theorem, it's obviously it's related to the diffusivity, and related to this constant times c, which is called the capacitance. And it's called the capacitance because it comes from physics and electrostatics. And Burke and Purcell were this who really started this field many, many years ago.
Mm-hmm.
And what they discovered was they did this very hand wavy, but they got the leading order approximation right. And what they discovered is that um, the total flux should be proportional to the diffusivity times the number of receptors there are, or NPCs or gates or whatever, times their radius, not their area. Now, if you look at the flux here, it's an integral over an area, so you might expect it to be area squared, but it's not. It's proportional to the perimeter. And that was quite a profound biophysical
Mm.
observation because it meant that a cell could take a small area, subdivide it, and create its perimeter, a very large perimeter, so that a cell could have a very small surface area devoted to all sorts of specialized receptors. But if their perimeter was large enough, then the flux would be similar to that of a all-absorbing cell.
Mm.
But there's no information in the spatial arrangement. It just gives you the leading order.
Mm-hmm.
And one of the first problems I did when I got into this field, this is a say this is a paper back in 2017 now, was to go back and using more sophisticated asymptotic analysis, first of all, was to give a systematic result, a systematic derivation of the Berg and Purcell result. And we showed that it's the leading order approximation in a sort of three-term asymptotic series, where there's three important terms. The first term is the Berg-Purcell result. The second term is a correction due to the spherical geometry, that the geometry matters, and that term comes in there. And the third is the competition term. Again, this Green's matrix, which we saw before in simpler problems, where it's a sum over all the different points of some interaction or energy. And that interaction energy is the following. It's a 1 over d, and it's got a logarithmic correction due to the geometry. Now, for physicists, this is very similar to the Thomson problem. What's the energy configuration of charges on a sphere? Um, but with a correction term in here for the spherical geometry. Now, if you look at this, you might ask, when is the flux maximized? Well, because this is a convex function, there's not a critical point here that suggests that they should have a preferred spacing. What this means is that you've just got to cram as many points on the sphere and keep them roughly equidistributed. They should be mutually as far away from each other as possible. And this maximizes flux. So you might ask, what sort of sets are good at maximizing flux and which ones are bad? Well, again, we want to know about the minimizers of this energy, this G here. And what we find is that uniform is actually really bad. Okay, so if you just take uniform points and slap them on a sphere, well, what you notice is that they cluster in that you get portions where there seems to be many, and then bits of the cell where there's none.
Mm-hmm.
Okay, so if you're looking at some experimental data and you use clustering, that could just be because the data is uniform. It doesn't mean there's some structure. So what we found is that uniform, and this is kind of, I think, well known, and this is actually why Monte Carlo methods decay so slowly, that this terrible 1 over root n convergence behavior. Why is it? Because when you start filling up spaces with random points, they leave
Mm-hmm.
big gaps, clusters and gaps. That's why Monte Carlo converges so slowly.
Mm-hmm.
And you can look at other various coverings which are synthetic, which are created to do a better job. And of course, you can use spherical coordinates here in the middle one. And there's
Mm-hmm.
a big literature in the mathematics and optimization and information theory, approximation theory about coming up with really, really clever coverings. Something very simple that you see on the right here is called this Fibonacci spiral, which is a very simple covering of the sphere, which does a really good job of being equidistributed. So something interesting here is about the role of homogeneity. If you see something homogeneous, then it's a sense that it's maximizing flux, but it doesn't happen by accident. What happens by accident is clustering, which does not do a good job of maximizing flux. Now, some of the things we want to do, we might not care about all this data. So a mathematical thing we can do with homogenization is say, well, we know there's thousands of these. Do I really need to know where they all are? Well, there's thousands of them. They have a small surface area. Yeah, there's 2% surface area. There's 2,000 NPCs. A typical cell might have 10 to the 5 receptors. Well, what can we do? Well, we can take this thing and do this mathematical procedure called homogenization, where
Mm-hmm.
we rigorously derive a uniform limit, where we take this absorbing, reflecting, and we derive from first principles the value of kappa, which you should plug in there to be consistent what you see. And what you see in here, F, is the absorption fraction. It's
Mm-hmm.
fixed. Sigma is the radius of the receptors. You see this square root correction, which comes from competition. And again, you see this log term, which is due to the spherical geometry. So the geometry plays a lower role. So it is important, but it plays a lower tier role, whereas receptor competition is the most important role. That's what this tells you here. So you got this Berg-Purcell result, receptor competition, and then the third role is from geometry. Now that was derived at steady state. So that's just looking at things in equilibrium. Sorry, this is just numerical verification. It was quite a tricky problem to numerically verify this, but we were able to do it. So it basically in the biophysical limits we're talking about here with 2,000 pores, it's accurate to about 0.3%, which is okay. It's okay. Now, one of the things that's very interesting is you do this process, you take all those data, right? Oh, sorry, all that microstructure, and you just replace it by a single number. Now, this is the exterior of a sphere, so we can write down a spherical heat equation. We can solve it exactly. We can calculate the surface flux. Now we can look at the full first passage time distribution,
Mm-hmm.
not just some steady state. We can look at the full distribution of arrival times. And here it is here. So in this example, it's a sphere with 51 patches. I'm using some triangulation of the surface here. And what we find very interestingly is that this approximation is fantastically accurate at representing not just the steady state character, but the entire time dynamics, except for, and I've had my cursor here, I would draw your attention to this right-hand side where you're seeing the relative error between the CDFs from the homogenized and the full numerical solution, whereas it does an extremely good job up till a very, very small window near very, very short times. So at moderate, or unless you have very short times, homogenization is this very powerful tool for averaging over microstructure to give you some emergent behavior, except for at very small times here.
Mm-hmm.
So let me just sort of,
Mm-hmm.
this is a vignette of some of the first papers I did when I got into this, and there's all sorts of details I'm missing, but what general things can I pass to you that hopefully are helpful when you were looking at data or working on things, and it's that uh, there's a first of all, if you're just interested in maximizing flux and you see some structure, you look at experimental data where things are very homogenized and equally spaced. Well, that's something that you can say right away that perhaps there's a mechanism here going on to optimize some sort of group sensing or flux, some group flux, or there's some sort of optimization problem happening here. On the other hand, if you see things that are uniform random, and a hallmark for this is clustering, where you see concentrations sometimes and uh, gaps, then that suggests there might not be an organizational mechanism, but it does on the other hand, lower flux. And homogenization is a powerful tool, and it can give the dynamics at all but the shortest timescales. So things I'm working on right now are general 3D surfaces. I'm going to report on this soon, and more complex reactions at each of these sites. And again, a lot of the numerics is quite tricky on this, so I'm having to we're really pushing some boundaries on computational PDEs to solve some of these problems. So that was what I talked about earlier is the forward problem. Okay, so we have an initial condition, we have a geometry and boundary conditions, and you go forward and solve the problem. But really, what the stuff we observe cells doing is kind of solving an inverse problem. So this is a paper I did with Sean Lawley and Chris Miles a few years ago, and it's doing this. It's solving a problem where now I get the fluxes at the receptors, and the question is, can I back out something about the initial condition? So now it's what if we have the fluxes at these receptors? How can we say something about the source? And we developed a sort of information theoretic way to answer this question. And I can tell you a little bit about what sort of results we got and what sort of implications they have. And the basic idea is that the cell has some belief distribution on where X is, that's P. And then once it gets some information at the receptors, it updates it to Q. And that change, the change in the belief distribution, there's a metric for this. It's well known in information theory. It's called the KL divergence. So it's the information gained from revising a belief distribution uh, from Q to P. Now, if the cell has no idea where the source is, then information should come at it just uniformly all over the surface. And that's the distribution here that you would expect on this. And of course, if you remember from calculus, this is just um, the measure on the unit. On the other hand, if you have exquisite knowledge of where the source is, if it's at a distance r above the North Pole, then the distribution on the surface is this function here I've given. And you can calculate the KL divergence from this. It's just a little integral here,
Hmm.
and you get this crazy formula. And what you notice is it diverges as you get close to one. Of course, I should say this, the sphere is radius one, and then the big R is the ratio of the distance to the cell radius. So there's this vertical asymptote at one, and the decay rate is R to the negative one half. So this gives you a theoretical bound on what you can learn at steady state through the receptor fluxes. And it's a theoretical limit in the sense that there's receptors everywhere and you have an infinite number of amount of information. And the key thing you learn here is this r to the negative one half is that if the cell is trying to detect a source that's twice as far away, then there's going to be 1/4 of the energy information that's available. Okay, so the scaling law here is important. So obviously it's harder to find more distant sources, and that rate is given by this theory, r to the negative one half, so a
Hmm.
quartering. Now, when we go to the practical problem where there's a finite number of receptors, um, we can do this for the finite. Again, we do this process we did before. We're solving uh, the steady state fluxes through each receptor, and we can construct these again versus using this Green's function asymptotic methods, and of course we can do it numerically as well. Um, and we want to ask the same question: is there any role for clustering? Does the receptor organization matter? And what we found is that, of course, just in reference to what I talked about before, that the best covering is a homogeneous covering. All right, so you maximize it, it's best in the sense of maximizing flux. So here is two cells trying to find a target just by looking at the steady state fluxes to the receptors. On the left, it's uniformly covered, homogeneous covering of 35 receptors. And you can see that the cell has a good sense on the distance, but a poor sense on the direction. So on the left-hand side here, the true source is the black dot, and this cell estimate is really just kind of, it doesn't have good sense of the direction. Whereas on the right, with the clustered configuration, the cell can form a much better estimate of where where the source is, but it's taking a flux penalty for this. So one possible role of clustering is to improve directional sensing at a cost of uh, a slight reduction in the flux, the total flux over its cell. Okay, so here's this process we have for taking the inverse problem for taking cell data, inverting out the source, and then using this, we can say something about the spatial arrangement of
Hmm.
the receptors in a principled way, from a first principled way.
Hmm.
So what sort of information do we get from this? All right, so in this information theoretic way, we can, we have a systematic but very mathematical way of trying to understand the information that a cell might have. And if it's using steady state receptor fluxes, kind of like the yeast problem, then it can infer the directionality of a signaling source. If that source is further away, it's going to be more difficult. And the rate is gives like it goes like an R to the negative 2 in the sense that if the cell is trying to detect a source that's twice as far away, it's going to have 1/4 of the information. Receptor clustering can improve directional sensing, but of course, as we discussed earlier, we know that clustering reduces total flux,
Hmm.
but not that much. By a couple, in that example there, it was like 5%. So again, one of the general directions I'm working on, I hope to report on this soon, is how to do this, extend this picture to cells that are non-radially symmetric. A lot of the stuff I'm showing you here is very highly specific to the sphere, where all sorts of nice things happen. That doesn't happen in general, and there's a lot of interest right now in how curvature affects these things.
Hmm.
Because we notice that when we look at experiments, that receptors seem to cluster at areas of pronounced curvature. And how do we give some explanation for why that might be happening? So that's the next vignette was the inverse problem, but at
Hmm.
steady state. Now, the steady state problem is kind of more familiar with the yeast. The yeast sitting there, it's got 2 hours to make a decision. It can suck in all the information at once. However, this neutrophil problem, it has to make a decision in seconds. And some of the results I showed you earlier, if you remember, the diffusion takes a long time to reach steady state. Okay, diffusion takes a long time. So there's some idea that perhaps the dynamics are important. So what I'm going to talk to you now is about how there's actually a lot more information in a very small portion of the signal, which is where homogenization fails at that very small part. So what I want to do here is go back to this neutrophil example and imagine that a source is released here on the left where that big red dot is. The molecules diffuse out and they have to reach one of those receptors in the cell surface. And the cell has this information. And we're going to go through this MLE process again, this process of using methods from statistics, using KL divergences and maximum likelihood estimates to take those fluxes and build
Hmm.
distributions for where the cell thinks the source is. And what you get if you do this is something interesting, and I've flipped it a bit here. So here's the cell. It's got some receptors on its surface. And on the right-hand side at t equals infinity, and what should I say here is I'm looking at what does the cell know at time t. So I'm starting at time 0. I'm going to go all the way to t equals infinity, which is steady state. But I'm going to take snapshots at certain times and ask, all right, what's the information the cell has at this time? Now, if we go all the way to infinity, we see this very broad, like we saw before, we see quite a broad distribution in angles, but quite a good distribution on distance. So the distance is quite well predicted. Now, as you creep back the time and say, well, what does the cell know? What's its best guess of where the source is at shorter times? What you see is that as time gets shorter and shorter, it's got a better and better guess on directionality. So in the left-hand figure here, you see it's got a very good description of where the direction is very certain at the cost of having a bad information on the directionality. So what we learned from this process is that actually that very small time signal where the homogenization fails, well, actually there's a lot of information on
Hmm.
directional sensing in that
Mhm.
small part of the signal.
Mm.
And the cell gets a hunch very quickly. So this is a
Mhm.
lot about decision making. And it's, well, maybe you should go with your hunches sometimes because the first information you receive about a problem typically has the most information. And we ask the question, well, what happens instead of looking at this as a continuous problem? What if we take this to the extreme and say, all right, if I release a bunch of particles, and I'm thinking about discrete particles now, I release a bunch of particles here at this red dot, m of them, where it's a finite number now, and I ask, okay, well, how long does it take the first one to get there? Okay, so now I've gone from continuous to discrete, so there's m particles that are my signal now. And I say, I'm going to take this to the max. If it's true that the short part of the signal has the most information, then what happens first one? So you release a bunch of particles at this red source here, they diffuse, they eventually reach the cell, and they have some time of arrival, and they have some angle of arrival. What's the error if the cell moves in that direction, theta 1? So suppose the cell just goes, the first bit of information I've got, I'm going to go in that direction. And I should say, I don't have some slides on this, but more and more, it seems that cells can solve this problem at very low concentrations, where it is truly a discrete number of reaction events at the cell which are giving the information that's required.
Mhm.
So let's talk about some of the mathematics of this. Okay, so let's say, suppose we've got these arrival times here,
Mhm.
and they're random variables, of course, and they have a PDF F, and they have a survival probability, which is like the complement of the CDF S of t. Now, what do we say about the distribution of the first arrivals? Well, this is what's known as a stream statistic, and it's the topic of this workshop I'm hosting next week in Chicago. And it says that, well, if there's all these things that are happening, all these IID processes, well, what's the distribution of the first one or the kth one? I'm not interested in the average anymore. Let's get away from the central limit theory. Tell me the distribution of the first one. Okay. Well, we can go through some processes here. If they're independent, we can split this up and we see that it's the survival probability raised to a power m. So one of the things that's very important is that uh, if we want to talk about extreme statistics, it's very important that we're able to obtain full first passage time distributions. At the beginning of the talk, I was talking about mean reactions times. Well, on average, if we average over everything, how long is this going to take? And in lots of examples, that's the quantity you want because there's so many things going on. But there's this class of problems where actually we're interested in one or two very distinguished events,
Mhm.
and the events that
Okay.
fertilization is another example of this. When sperm chase an egg, who cares what the average time it takes to get there is?
Mhm.
It's the first one that fertilizes the egg, not the average one. So there's many processes in biology that are governed by this principle, and it's kind of a very big topic at the moment, how this shapes the function of many biological systems.
Mm.
So what does this do? It has a sharpening effect. So I'm
Mhm.
going to choose just a toy distribution here. You can see this f here. It's t to the negative 2 e to the negative 1 over t. It's a toy distribution, but it's very similar to those that appear in diffusion processes a lot, which are basically of form t to the p e to the negative c over t. And I chose this one because it's very easy to calculate the survival probability here. And what you see is that the PDF itself, f, has a very broad distribution. Okay, so that's the blue curve there. This is one the time it takes one particle to get there is extremely broad. However, the first time, if there's n of them, if there's 100 of them, and that's the yellow curve there, it starts looking like
Mm.
it's a very tightly distributed quantity around a good mean with a tight variance. So what you're seeing is that the mean is becoming more and more deterministic. And I mean it in the sense, I mean deterministic is that it's going to a very fixed mean with a very small variance. And this is classic statistical modeling. And there's a beautiful theorem. It says you don't need to know anything about you don't need to. For any IID process, this will converge to one distribution. So back here, what did I say? I said the times are just IID random variables with some PDF and some CDF. theorem from extreme value theory tells you that you will always converge to one distribution. It's got a shape parameter gamma, and there's different characterizations. It's called Gumbel if this gamma is zero, it's called Fréchet if it's positive, and it's called Weibel if it's negative. But you basically go to one PDF which describes this, and it's an exponential of an exponential, basically.
Mhm.
And so we can use this theory to go back to this problem and say, okay, what's the error in moving this direction? And I mention again that we have to get the time distribution first. Okay, so perhaps you see this is that the particles that take a long time to get there are going to be more averagely distributed along the circle, whereas the short ones go very quickly. So we have to get the times first, and then from that we can calculate the angular distribution. And again, pages and pages of math later, and all sorts of classic methods from applied mathematics, Laplace's method, blah, 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 blah. What's the answer? Okay, so if you go on the first particle, your
Mhm.
error, you take the 5, your error will be normally distributed, and that's because of the symmetry of the problem, that it's normally distributed against the true answer. And your error has variance, and here's the quantity here, r times 1 over r minus 1 squared over log m, where log m is the number of particles, and it's a proxy for concentration. So the larger the concentration, of course, appearing on the denominator, we see that it's going to be more accurate, more tightly bound around the true value. Obviously, at r equals 1, this goes to 0, signifying that if this source is very close to the target, of course, it's a very easy problem. But crucially, as you get far away, what about the large scale problem? You now get r to the negative 1. So by using extreme statistics, by ignoring the later particles and only looking at a small number of early particles, you've changed that r to the negative 2 to an r to the negative 1. So you'd be 4 times as tough to twice as tough as the source is more distant.
Mhm.
And if I take these experiments here, I go back to these experiments and say, and if you look in the caption of the figure of this experiment, it says, look at, we saw something at 2 seconds. You take some numbers that are, and you see that the time that we would expect from this theory is about a second, depending on what these values are.
Mhm.
And therefore, it serves as a theoretical minimum bound on the response time that a cell has. So what this tells you is that the first particles to arrive at the cell take about a second, and then the experimentalists know that the reaction of the cell is about a second later. So this serves as a theoretical minimum bound on the reaction time, which neutrophils are pretty close to. So it's evidence that they're reacting way before steady state. They're taking a gut hunch, part of the information, and that's the way they're moving.
Mhm.
So let me just summarize this section and leave my time to get onto the last part.
Mhm.
And that is the directional information is front loaded in the earliest signal to the cell. And I think this is a broad phenomena
Mhm.
around a
Mhm.
lot of social decision making. Like we kind of go with our gut. And the evidence I'm presenting here is saying that in diffusion processes, that's a good idea because the later information has way less quality. So to reach steady state, you have to integrate all the information that came preceding. You take the high quality information and you average it with the low quality information to get something that's just okay. Taking the early part just gives you the high quality information.
Mhm.
And the times, there's two components to this. There's the times, which seem to encode the distance information better, whereas the locations of impact encode the directionality. Now, all of this mathematical analysis is based on MLE and asymptotics and solving PDEs, so it's no guarantee that cells actually do this
Mhm.
because MLE requires quite a lot of computational capacity. You have to store all these things, build your likelihood, and send it to computer. However, there are some principles here that we do see is that simple things based on a small number of estimates can be highly accurate if you choose the right one.
Mhm.
And we've got there's some natural things that went on from this. And we actually just have a preprint out that's, well, what about if we look at multiple arrivals? What if the cells have access to times, or maybe they just have access to the inter-arrival times? We can say a little bit more about the information you would get from taking multiple times or some subset. And of course, natural questions that we want to answer is when does this strategy fail? And what is known generally is that these strategies work for single cells. Then, as you get multicellular, or there's multiple sources, or there's a very complicated environment, then these simple principles start to fail and the cells have to cooperate together. So there's a whole next extra field of collective sensing.
Mm.
Uh, and there should be some trade-off at a certain critical density where the methods I've discussed here don't work anymore because there's too much noise. Okay, so let me just conclude here with this part talk. And I've talked about forward problems, some of the things we can say. And of course, one of the main things is how do we tie together geometry, the location of all these sites.
Of
And
course.
the second thing I talked about was using this information to solve inverse problems that the
Mm.
cells are actually solving it, and we see them solving in experiments. That's what we see. We see them solving inverse problems. And just let me just go straight to this second slide. I hope you don't mind. I'll just try and be as quick as I can here because, of course. And this is more very recent work on two talks. So this is two papers here. Um, one of which appeared in SciAm Applied Math, and another one which is just about to appear in PRL. And
Hmm.
it's again about this problem. A lot of what I talked about is idealized. If okay, well, what's actually inside a cell? It looks like this. On the left-hand side, you see that the microtubules, these linear structures,
Hmm.
which guide the diffusion. And as I mentioned before, diffusion is a very weak principle on its own.
Mm.
And on the right-hand side, this is more of an ecological example. Some of my collaborators have come with a lot of ecological problems, and a lot of the mathematics is the same. What you're seeing here, and maybe this actually is something similar you see in your tracking data, because it's the path that a whale is taking from its feeding grounds to its mating grounds, and it looks like a random path, but of course you see it has some guidance. So it's a random path, but it's not a homogeneous random walk. This is not, there's no way this is diffusion. So how do we sort of go a bit more sophisticated and talk about first passage time processes? Try and answer the same question, but with environments where we know there's cues or spatial features. And the framework we're doing this is called the velocity jump process.
Mm.
So it's a generalization of Brownian motion. It's a type of random walk
Hmm.
where instead of drawing your new position from a normal distribution like Brownian motion does, it says it's piecewise deterministic. And it says we're going to go along deterministically in some velocity v, and then at some rate mu, we're going to jump to a new velocity. And that velocity could be continuous or it could be discrete. So on the left-hand side, you're seeing two particles. The yellow particle draws its new direction. This is the left particle, the left picture is the slow, it's mu equals one, so that's slow switching. And you see that the particle is drawing its distribution from a continuous angle between 0 and 2 pi. The speed is the same, so it's only directionality. And then the purple particle is doing a snake. So it draws from a discrete distribution
Hmm.
based on four numbers, 0 pi
Hmm.
over 2 pi or 3 pi over 2. On the right-hand side, you see a faster switching rate. Okay, so things are switching a lot faster, but it's the same process. So this is a piecewise deterministic system. And the key things here are the turning kernel, K. So it says that if you make a jump at position X and you're at velocity V, then
Mm.
you'll go to V prime. So what's the probability of you going to a new position? And I'm thinking about these very spatially, but I think a nice way to think about this is also in decision-making processes. What's the probability of me changing my mind on some topic, given that where I am in state space? Now, I'm going to give you some results on this, and I'm going to go quick, because otherwise I'll be out of time. But one of the main assumptions that we need now, and of course it'd be beautiful to repeal this, is that the turning rate does not depend on your velocity coming in. Okay, so you have some, then that, we can talk about that assumption, but this is kind of what we need to push through our theory. And I'm
Hmm.
going to just write down the result that we have here for the mean first passage time. Okay, so
Hmm.
what I want to know, I'm back to mean first passage times here. So suppose it doesn't matter your incoming velocity. If you jump at a position X, you will go to some distribution Q, where your velocity will be drawn from this distribution. Then we have this, and it looks gnarly, but I'll just say it's an integral partial differential equation. and it's of tensor type. It's an anisotropic diffusion equation. And this looks horrible to look at, but let me talk you through some of the things in here. The differentiation part is comes just from space. So the spatial information comes through derivatives. And the integration part of it comes
Hmm.
through the velocity space. So just to give you some idea, why is that an integral differential equation that we get here? Well, we know it's a spatial process. We're used to this from Brownian motion, but now we have this velocity consideration, and that makes it an integral velocity PDE. So one of our new results is that we've derived this PDE for the first time ever. So we've known about mean first passage time theory for Brownian motion for many years. This is the first derivation of a mean first passage time equation for a velocity
Hmm.
jump process, and it's an integral differential PDE. Now we can do various analysis on this to make it simpler. Okay, so one of the things we can do is look at the parabolic approximation, which says that, look, let's just look after many, many jumps. Let's look at some process where we've done many, many jumps and we've lost information about our initial velocity. And now we get down to this anisotropic PDE here. It's still a tensor type. Okay, so it's anisotropic in the sense that it depends on space through the diffusivity, but it also depends on the cross derivatives. So the diffusivity is in the x1 and x2 directions.
Hmm.
Now, what sort of turning kernels are we interested in? Well, one of the main things is linear filaments. So suppose you've got a microtubule and it's got a directionality which is given by a unit vector gamma. Then what might happen is a particle gets close to the
Hmm.
microtubule, and I'm looking at the
Hmm.
picture right here. Then when it binds to it, if it's unbiased, then it's got equal chance of going to the right, equal chance of going to the left. And this would be the distribution of your Turner kernels. This is called the von Mises concentration. So what you see here is that it's essentially a Gaussian distribution, but on a disk. The usual Gaussian is negative infinity to infinity. This is the Gaussian on 0 to 2 pi. Now I can change these numbers, these beta parameters here, and I could make it biased. So here, if a particle, if a walker or a protein was to get to this tubule, it would have a much larger chance of going to the right when it arrived there. So when we take a kernel like this, we can calculate all of these messy coefficients, and they're in terms of Bessel functions. So we can calculate both the diffusivity tensor and the drift tensor in terms of modified Bessel functions of type 2, 1, and 0. And it's a messy object here, but just that we can map this all down to, and let me just, sorry, pardon me. We can map this all down to these two parameters, these two functions. So when can we actually solve all this analytically? Well, we've done some calculations for the unit disk, and there's two choices that are natural. One is radial alignment. So what you're looking at here on the left is, suppose you have a particle undergoing this process I talked about, this velocity jump process, and every time it jumps, it has a preference to move on the radial direction. Okay, so a particle starts, and now all it wants to do is it prefers to move radially. So at any point where it decides to jump, it says, what's the radial vector to the origin? And I'm going to move in that direction. And you see that. You see that each of its paths are essentially straight lines emanating from the origin. Whereas on the right-hand side, we do the tangential thing. We move from at each point, the particle has a propensity to move along the tangential vector. And of
Hmm.
course, it goes through this random walk where it's doing these big spirals. So all that's happening here is you have a particle that's moving in a straight line at some exponential rate. It decides to change direction. And when it makes that change in direction, it decides to have a preference along the tangential direction. And you get these beautiful spirals. And we can boil ODE in this case. For this particular case, if you're looking at these radial directions, we could boil different down to an ODE. We can solve this ODE. I'm going to go a little bit faster here because I can tell that I'm out of time. We
Hmm.
can solve this. What you see is something different, is that the exponent on the solution changes. So if you know a little bit about ODEs in a disk, this fundamental solution is a log. Well, the fundamental solution changes to this factor here. And since I'm going a little bit ahead of time, I'm just going to just give you a vignette here. We took some ex images here and we extracted the linear data from them, and then we solved the PDE on these data-driven sets, and we mapped it to particle simulations where particles are diffusing, but they're influenced by these linear structures. And it all matches up, the theory matches up, and we can do this for any sort of. So this was previously on how to give what's the theory for the mean first passage time for velocity jump processes with these complicated queues, and we can sort of write down a theory from this. And since we've got the operator, the infinitesimal operator, we can also write down a Langevin equation for this. So that means that we can map the whole thing to an Ito SDE, and that allows us to actually go beyond mean first passage time statistics, and we're able to uh, again say something about full passage time statistics. So we can go from a piecewise deterministic system to
Mm-hmm.
some homogenized PDE of an integral differential equation, and then we can take it the next step and derive a continuous SDE. And then people can use Fokker-Planck equations and whatnot to study this as well. So
Mm-hmm.
this is something that takes microscopic decision-making processes and
Mm-hmm.
gives you a pipeline to take it from PDEs to SDEs and and all the way through. Okay, so I will end there. And I'm just
Mm-hmm.
at the end of my time, a little bit long,
Mm-hmm.
but thank you very much for listening, and I'm happy to answer any questions.
Thank you, Alan. Thanks so much. Okay, so let me stop the recording.